Don't forget to like, subscribe and share the videos.
First news is Nobel Prize in Medicine and Physiology. Okay, so who has received this prize? What is its significance and for what invention or discovery? Let us understand that. So the Nobel Prize in Medicine or Physiology for 2023 has been awarded to Kathleen Carrico and Drew Weissman for what? For their groundbreaking work on nucleoside base modification. So they have done nucleoside base modification of what? Of messenger RNA, shortly called as mRNA. So we need to understand in detail about what is mRNA and what is the role of mRNA vaccine. That we need to understand in detail, and how have they modified this nucleoside bases and why did they modify nucleoside bases. All these details we must have to understand in detail. Okay, so the discoveries by the two Nobel laureates, okay, this was critical for developing effective mRNA vaccine against COVID-19. Yes, okay, so that is the reason they have been awarded this particular Nobel Prize. Okay, and this pandemic was there in early 2020. Okay, it started in early 2020. So this method was very useful in developing effective mRNA vaccine against COVID-19. Okay, so let us understand further more details. So we need to understand the challenge that they had and how did they face the challenge or address the challenge. Okay, so generally cells possess an inherent capability to detect foreign materials. This is there in every cells. Okay, and there are particular cells called as dendritic cells, okay, which play a critical role in immune system. So these dendritic cells play a very critical role in our immune system, and they have this ability to recognize in vitro transcribed mRNA. So this term we need to understand. What is in vitro transcribed mRNA? And this dendritic cells are going to recognize these in vitro transcribed mRNA as foreign, which means they are foreign material to these cells. Okay, which means they will be creating antibodies against these foreign materials, but then they are setting off an inflammatory response. Okay, generally in vaccines such inflammatory response are undesirable. Okay, so this reaction could potentially lead to harmful side effects and undermine the vaccine's efficacy. Okay, so when in vitro transcribed mRNA is going to be used for vaccine production, then these side effects like inflammatory responses could potentially be a threat to human beings. Okay, furthermore, another challenge that stemmed from the fact that in vitro transcribed mRNA was highly unstable, one thing, and then they are also susceptible to degradation by enzymes. Okay, so they are highly unstable, they are susceptible to degradation by enzymes, and they are also causing inflammatory responses. These are the challenges that in vitro transcribed mRNA had, and how did they address this challenge? Let us understand that. Before that, let us understand in detail about this in vitro transcribed mRNA. What is it? In vitro means in lab. Okay, vitro means glass. Okay, so they are going to do this in test tubes. That is why it is called as in vitro. Okay, so in vitro transcribed mRNA is a type of synthetic RNA. So it is synthetic RNA which is produced in the laboratory. Okay, so a question based on this may be directly asked in your prelims examination. What is in vitro transcribed mRNA? Okay, so it is produced in laboratory by using DNA template and RNA polymerase. Okay, so generally RNA is produced from DNA, right? So it is produced by using a DNA template and RNA polymerase enzyme. Okay, and this in vitro transcribed mRNA can be used for various purposes like making RNA probes, vaccines, and also for production of protein. Okay, so these are the details which we need to understand with respect to in vitro transcribed mRNA. Furthermore, details that we have to know is the discovery made by these two scientists who won Nobel Prize. So Carrico and Weissman observed that dendritic cells identify this in vitro transcribed mRNA as foreign, which we already saw, and activating them and causing release of inflammatory response, okay, inflammatory signals. And they questioned why this mRNA was considered foreign, why this in vitro transcribed mRNA is considered as foreign, whereas our mRNA from mammalian cells, in human beings we have mammalian cells, in mammalian cells of mRNA such response is not there, okay, so which did not trigger the same response, okay. So why is there this distinction between mammalian mRNA and this in vitro transcribed mRNA? They were researching this. And mammalian cells, what are mammalian cells? Let us understand that also. Mammalian cells are eukaryotic cells, okay, we all are eukaryotic organisms, okay. So eukaryotic cells that belong to animal kingdom and have a nucleus. Eukaryotic means it will be having a well defined nucleus and other membrane bound organelles. That is the meaning of eukaryotic, which is the opposite of prokaryotic, which will not be having a well defined nucleus and membrane bound organelles, okay. Next, this led them to realize that there must be a distinct property separating the two mRNA types. So what is the distinct property that we need to know? So what was the breakthrough? RNA, like DNA, consists of 4 bases. We all know adenine, guanine, uracil and cytosine, okay. In DNA the 4 bases are adenine, guanine, thymine and cytosine, okay, whereas in RNA it is adenine, uracil, guanine and cytosine, okay. Here these two scientists have noticed that the natural RNA from mammalian cells often had chemical modifications in its bases. It is just because of these chemical modifications they were not giving such inflammatory responses, okay. So that is what they have found, that is their finding, okay. So they have hypothesized that absence of these modification in lab made mRNA, that is in vitro transcribed mRNA, might cause inflammatory reactions. So the point that we must have to note here is these mRNA, that is lab made mRNA or in vitro transcribed mRNA, having absence of these modifications in bases. What are the bases? Adenine, uracil, guanine and cytosine, okay. And to test this, they created various mRNA variants with unique chemical alterations and delivered them to the dendritic cells which were showing the inflammatory response, okay. And their results showed a significant reduction in inflammatory response when base modifications were included in mRNA. So for this nucleoside base modification only they have been awarded Nobel Prize, okay. So this we need to know. Next is this discovery transformed our understanding of how cells recognize and respond to different types of mRNA, with profound implication for mRNA's therapeutic potential, that is its potential as a vaccine, okay. And their subsequent studies in 2008 and 2010 demonstrated that mRNA with base modification led to increased protein production. This is also there, okay. So these two scientists have research, their research removed the critical obstacle. What was the obstacle that we had? That these mRNA were producing inflammatory response, okay, and making mRNA more suitable for clinical application. So this is the background which we need to understand. Next is mRNA vaccine and their working. How does mRNA vaccine will work? This we need to understand from exam point of view, from prelims and mains point of view, okay. So how does mRNA vaccine work? mRNA stands for messenger RNA, we already know, but what is messenger RNA? It is a molecule that carries genetic information from DNA to the protein making machinery of the cell. So it is like a messenger, it carries genetic information, okay. It is sending the information from DNA to the protein making machinery, okay. So mRNA vaccines use synthetic mRNA that encodes a specific protein from a pathogen. So in case of this coronavirus, here the protein is spike protein. In coronavirus, you would have noticed that it will be having spike protein. Just because of these spike, It is called as corona. This shape is called as corona. This spike shape is called as corona. Okay. So these spike protein are encoded, and they will be, that message or that genetic information will be transferred to the cells. After it being transferred to the cells, when the mRNA vaccine is injected into the body, some of the cells will take up the mRNA and use it to produce protein. RNA's job is actually producing protein. Okay. So it will use it to produce this protein, this spike protein, and the protein then triggers. So a foreign agent has entered into the body. So whenever a foreign agent is going to enter our body, then there will be an immune response which produces antibodies and memory cells, and there will be memory cells that can recognize and figure that pathogen in the future. Okay, so this is how the mRNA vaccine works. Okay, so let us see that in the picture. mRNA coding for SARS-CoV-2 protein, spike protein. Okay, here it has been coded, the code is being injected. Okay, that mRNA, the messenger RNA, is being injected. Here the mRNA released into the cells where it is used to produce spike protein. Okay, these spike proteins are going to be produced inside our body, and to that our body will be producing antibodies. Okay, so creation of antibodies that can recognize this virus. So once antibody is created, it will go and it will go and defend this spike protein, and that memory will be stored in our immune system. Okay, so after that, whenever the spike protein is encountered by our body, then automatically our immune system will react to it. Okay, so this is how it works. This vaccine works.